have a nice fish on your screen now?
Wonderful, beautiful fish.
Yes. And this fish in Faroese is called gull-laks, which means golden salmon. And the main part of my talk will be on this one, but I will also talk a little about general about our work and our participation in ERGA. This beautiful fish here is one of the pilot species in ERGA. So how did we get there? So let me first present the Faroe Islands to you. So we are in the middle of the Atlantic Ocean, so kind of halfway between Scotland and Iceland, and Norway for that matter. And as you see, it's a small piece of land surrounded by a big sea. The population in the Faroe Islands is around 54,000 in total. And this is less than many of the European universities. So it is a small nation. It is a self-governing nation, but it is under the Danish crown. And this sometimes makes a bit confusion because, for example, Denmark is an EU member, while Faroe Islands is outside EU. It has a university, around 1,000 students in total. The major part is studying nursing and pedagogy, but also biology, where we have both ecology and molecular lines. It is 18 islands, 17 of them are populated, a little less than 1,400 square kilometers, and a lot of sea, 261,000 square kilometers for the economic zone. It is probably no surprise that the main industries are fisheries and aquaculture. And it has formally what is known as a subpolar oceanic climate, but more informally, it is called bad weather. Lots of bad weather. So how did we come into ERGA? So as Alice said, we had already sequenced the Atlantic herring, and we had performed the population genomics on the stocks. We had also tried to sequence silverfish, which is not a fish, but a primitive insect. It did not succeed so very well. But in that part, we in fact detected that this guy here was also present in the Faroe Islands. It had not been registered before, but in fact, it was quite common. And personally, I had the pleasure of having both of them running around in my house. I got to hear about what later became ERGA by a mail from Professor Tom Gilbert, Copenhagen University, in August 2020. And I immediately jumped to that initiative. And we started to apply for money. And in June 22, we got our first positive answer for the project that we called Initiation of the Genome Atlas of Faroese Ecology. And the focus on that project was on the greater silver smelt and lesser sand eel, which both are then pilot species, Faroese pilot species in ERGA. But we did not get any money at that time because we would not get the cash paid out before the project was fully financed. So we just had to continue applications, and it took another year before we had the money and we really could start. The aims of our local genome atlas is first, the same as ERGA, to acquire high quality genomes for all eukaryotic species in the Faroe Islands and in Faroese waters. But in the Faroe Islands, we'd like to have such things being applied as soon as possible. And for many purposes, population genetics is something that you can apply and you can build on these reference genomes. So that is included in the part on this greater Argentine or greater silver smelt, as it is called in English. We have a third aim also, and that is to try to engage the public. And we want to establish an information data bank for Faroese species, all Faroese species. And we should combine that with a citizen science registration database, something similar like other Nordic countries at least have. And we hope that this can increase the interest among the public for biodiversity and taking care of nature and natural values. So then to the greater Argentine or greater silver smelt. This fish here, this is the one that now has been sequenced by Sanger and Mark Blackster and company. And it was caught in Faroese waters the 12th of September 2021. And we are building the population genetics on this genome that was established from this fish. Even if the greater silver smelt has been commercially fished for maybe around 50 years, we know far less about the stocks of the greater silver smelt than we do of, for example, herring or mackerel or cod. What we do know is that it grows quite slowly. It matures at 6 to 8 years on the average, but the range is 4 to 12 years. So it is a fish that becomes mature quite late. It can become very old. The oldest registered fish that I have seen is 36 years old. But the main catches of the fish that is relatively young fish. It's 6 to 10 years old. So it is just at the time when they start to mature. And the curves that we see here on the right are taken from catches in 2014 and 15 from the area around Faroe Islands and Scotland. But the curves are similar in the whole Northeast Atlantic. And it doesn't vary much from year to year. The greater silver smelt does not seem to have specific spawning grounds. At least we are not aware of them. We don't know them. And additionally, it has a very extended spawning period from relatively early in the spring to quite late in the autumn. And that is something that is quite different from the different stocks of herring and cod and mackerel. So what we do not know about the greater silver smelt is, for example, whether there is a single population in the Northeast Atlantic or there are many local populations. Just hiding the phone because it is screaming a little bit at me. ICES has divided the Northeast Atlantic into 4 management areas that we see with these different colors. One around Iceland and Greenland, one around Norway, one around Faroe Islands and Scotland, and one, should we call it, European and Mid-Atlantic. But we do not know whether there is one population in the Northeast Atlantic, or there are several populations that follow the management areas of ICES. In fact, when we talk to the local industry, we got to know that fish that is caught just here west of the Faroe Islands, in fact, are different from fish that are caught just south of the Faroe Islands. So that is an indication that there could potentially be quite local populations, but there is no real data for that. So we also then, of course, do not know whether each population are having specific spawning grounds, or they more or less randomly mix throughout the Northeast Atlantic. So it could be like this, but we don't know. And related to that is there a gene flow between the different geographical areas, whether it is adult individuals that actively swim from one area to another, or it is egg and larvae that passively follow the sea currents from one area to another. If it is passively following the sea currents, we should expect certain patterns of connections. So we don't know whether there are such connections like this. So we will look at DNA, which, no surprise to you, can partly, or maybe for some of these questions, fully give us answers to these questions. And to do this, we have collected samples from Gator silver smelt. The red stars here are indicating areas where we already have got samples. 2 samples from Iceland, or sampling areas from Iceland, and 3 sampling areas from the Faroe Islands, 1 from the northern part of Norway. And we hope that now in March we will have 2 more samples from the southern part of Norway. And then we have also a sample from the Porcupine Bank outside Ireland, and that we have got through Spain, the Spanish Institute for Oceanography. So what we will do in this part of the project is to do low coverage sequencing of around 50 fish from each area. And we are going to compare the genomes from all these individuals within each of the areas to find identities and variations within each area. And we are going to compare all the areas to find sets of variations that hopefully it could be specific for each area. And if they are specific for each area, it's a clear indication that in fact there are specific subpopulations. And this knowledge then can be used in the management of the greater silver smelt in the Northeast Atlantic. So ICES may change their management areas, for example. And this will help to secure sustainability in the fisheries. And as probably many or all of you know, sustainability in fisheries is something that is quite important. We have, for example, the collapse that happened with the cod outside Newfoundland in the early 1990s, after relatively good catches through the 1980s and relative very high catches, record high catches in the 70s and 60s. but then it suddenly collapsed. Similarly, there was also a collapse of cod outside Greenland in the late 1960s, early 1970s, after record high catches just a few years before. But here we have the underlying biomass, so the total stock, and we see that this was decreasing quite a lot during the years of these record high catches. And then we have the Norwegian spring spawning herring, which had record high catches at the end or middle of 1960s, and then within a couple of years it collapsed. And again, we have the underlying biomass, and we see that this decreased in the 10 years before this collapse. So just relying on catch numbers or landings for the fish is something that is quite dangerous. It is not sustainable to only rely on catch numbers. So we can also use the results that get out from this project to develop tools, for example, for tracing fish and fish products. This is something that could be of interest for the local industry. And also in the fight against illegal, unreported, and unregistered fisheries. And in fact, we have a twin species of the greater silver smelt, which is in Faroese called Stovnsgull, the lesser Argentina, lesser silver smelt. And here, the lesser silver smelt is the fish at the bottom, and the greater one is at the top. And these two fishes, these two individuals were caught in the same trawl hall at the 9th of August last year. So there is absolutely a possibility of mixed catches. So we can also use this data that we get out to develop tools that can assess mixed matches. And this lower individual here, the lesser Argentine, that individual is now being sequenced in Copenhagen. So hopefully we will get out a sequence, a good sequence from this one also. So this can help us to get the tools that, for example, can tell whether there are mixed catches, whether a fish comes from one stock or another stock. So this can be quite helpful. But we can also use this knowledge for more general information and more basic biology, for example, to tell us about what are the genetic characteristics of the order of Argentiniformes, which both of these fishes belong to. And these are the first genomes from this order. No genomes from this order have been available before. And we could also potentially, if the population genetics become good enough, say something about which route the greater silver smelt followed into the North Atlantic after the last ice age, because it was probably much too cold in the ocean in the Northeast Atlantic during the ice age. So they probably came in one way or another, maybe from the European side, maybe from the American side. We don't know yet, but maybe we can tell that from the data that we get out. So it is a lot of people that are involved in this project and also in other projects, that sub-project of our local genome atlas. There are also other species in the pipeline or are planned. And these people and these institutions have contributed with in-kind contributions. But of course, without cash, we are not able to do much. So these are the institutions and companies that have been able to supply us also with cash money for this project. And with this, I would just say thank you for your attention.